Okay, hello girls, let's look at 1.3, communicating in biology, and that involves tables, graphs, and biological drawing. So the first one is table. What do you need to take note? From the table here, you can easily identify which one is the manipulated variable and which is the responding variable. Okay, by looking at the left side and also the right side. So this is the table. On the left side is the manipulated variable. Okay, and on the right side is the responding variable. Okay, moving on, let's look at graph. Okay, for graph, this is called the y-axis. Okay, and this is called the x-axis. So very easy to remember RM, Ringgit Malaysia. So R, responding variable, okay, is represented by the y-axis, whereas the M, manipulated variable, is represented by the x-axis. Okay, second point is that the scaling, skala, pemilihan skala. So scaling on the axis must be uniform. Uniform here means pemilihan skala itu mestilah seragam. Okay, for example here 10, so each section here is 10. Okay, the third point is that you should be marking the points with appropriate symbol, which is X. Okay, and then you use a sharp pencil. Jangan gunakan pensel yang tumpul, dan juga elakkan gunakan dot. Okay, sebaik-baiknya kamu gunakan tanda X. Gunakan pensel yang tajam. Okay, and then when you're joining the lines, also make sure that it is a smooth line like this. Okay, no extrapolating, tidak perlu mula dari sini, kosong, dan tidak perlu lanjutkan. So it must be on the dot. Okay, and then the last point is very important also, where you need to write the graph, the title of the graph. Okay, so how to write? It is graph, then you put here volume of oxygen release, and that is the responding variable, versus manipulated variable, in this case is the temperature. So you get volume of oxygen release versus temperature. Then that will be the title, and you must write it on top here. Okay. Now this is the one. Okay, next is the bar chart. So this is called a bar chart. Okay, very important points that you need to remember is that they are not attached to each other. Jadi ada jarak sikit di sini, mereka tidak bersambungan. Jangan letak B di sebelah A dekat-dekat. So mesti ada jarak. Okay, dan juga make sure it is also uniform. Okay, ini adalah histogram. So in the histogram, you see the difference just now. For bar chart, there is a gap, but for histogram, there are no gaps. So they are close to one another. Okay? Okay, let's look at this very important section of biological drawings. Sebenarnya ada magnification power as well. Okay, let's look at the characteristics first. Mula-mula tu mesti besar. For example, it is your A4 paper, at least half or 75% of your A4 paper. Large and accurate, sharp pencil, bukan color pencil, and definitely not pen. Okay, number 2, not shaded artistically. Maksudnya, kamu jangan buat tone. Macam subjek pendidikan seni kan, kadang-kadang kamu buat satu, kamu gunakan pensel untuk buat toning. So that is not allowed in biological drawings. Okay, so the drawing must be clean. Okay, the lines must be clear and continuous. Okay, jangan putus-putus, sebab bila kamu melukis, kadang-kadang kamu buat toning, artistic drawing, the lines will be not continuous, right? So you must avoid that. Okay, do not use a ruler to draw the outline of a specimen. Biarpun contohnya sel tumbuhan ada bentuk tetap, okay, but you must use freehand, draw a line like that without using a ruler. Okay, this one very important. You must label all the drawings. The label lines must point to the structure. Okay, no arrowheads. Jangan gunakan anak panah. Contohnya yang ini adalah gambar. So, bila kamu melabel, mesti tunjuk betul-betul. Contohnya di sini cytoplasm. Dan jangan gunakan anak panah. It must be straight. Okay, and not cross each other. Jangan buat, let me see, like this. So the labels, the lines are straight. There's no arrowhead. Jangan ada anak panah. The drawings are clear. The lines are continuous. So this is detailed drawing, and whereas this is plan drawing. So this is how you should be. You should practice. Yeah. Make sure the lines are not crossing one another. For example, you want to label the pith. The cortex is over here. So you don't do the cortex, put it on below here. So your lines, your label will go down and it is crossing the labels of the pith. Ini maksudnya. Not cross each other. Your drawings must have titles, very important. Okay, you do not assume that the people who see your drawing will understand what is that. So it must have titles. So each structure in the drawing must be placed in correct position. Its size should be appropriate to the rest of the structure. Okay, posisi betul. Maksudnya, you see that the phloem is at the outer. Okay, xylem is inner. Okay, and you have the cambium over here. So posisi mesti betul. And then the size should be appropriate, proportionate. Maksudnya kalau kamu lukis phloem and xylem over here, it shouldn't be too big. Jadi dia punya size tu adalah bersesuaian dengan gambar raja keseluruhan. Contohnya kalau kamu lukis nucleus, the nucleus shouldn't be very big. Okay, maybe just a suitable size. Okay, a plan drawing is line drawing that shows outline of the structure, detail. This is detail. Okay, magnification factor must be stated. So ini 2 kali. That means the length of the drawing, kamu lukis dia besar sikit kan, but the length of the specimen is actually 1.2 only. So there's a magnification power of 2 times. Kalau structure itu adalah besar, but you lukis dia kecil sikit, so should be in the form of pecahan. But usually, kalau kita lukis benda ni, we are observing using microscope, right? So, selalunya dia adalah kuasa pembesaran. So, you will get a whole number like this. Okay, let's look at sagittal, frontal, and horizontal planes. Sagittal. So, yeah, this is sagittal. Divides the body right, left. Frontal. So, this is frontal, divides the body front and rear. Rear means belakang, ini depan. Horizontal divides the body into upper and lower parts. Okay, next. Okay, you should be familiar with cross-section and also longitudinal section. Okay, ini memang senang ingat. Ini keratan rentas. Cross-section, you cut like this. Merentas tubuh lobak ini. Whereas if it is longitudinal section, keratan memanjang. Okay, direction. Direction. So let's look at the terms. Superior, lateral, tepi. Okay, inferior, di bawah. Anterior for fish. Okay, posterior, ventral dekat bawah, and also dorsal up. So these are the definition of the terms. Okay, be familiarized with the directional terms in biology. Okay, the last part of chapter 1, that is 1.4 scientific investigations. I think you should be familiar with this since you have been doing it since primary school. Alright, report writing. So you have to have the aim. The aim is that you have to change it. You must start with to study the effects of an activity on the pupil's pulse rate. Okay, then you will have a problem statement, and this is in the form of question. So you should end with a question mark. There's a hypothesis which will relate the responding and also the manipulated variables. Okay, there's a fixed variables over here. So 3 pemboleh ubah ini. Materials and apparatus. Okay, specimen, bukan semua eksperimen ada specimen. And procedure. Procedures is look at the kata kerja. It is in front, you see. Count the pupil's pulse. Okay, how is it counted? It's asked to carry out, but it depends on the experiment. Usually it is in active form, but when you write a report, you should change the procedure of the one given in the lab manual to passive form. Is counted, is asked, is carried out. So the results is recorded in the following table. Okay, keputusan. So remember just now, MV and RV, which is responding, which is manipulated. Vigorous and activity manipulated. Okay, vigorous activity, and then the pulse rate. So you're recording the pulse rate. So jadual ini lain sikit lah. Okay, kena biasakan. Then you will have discussion, conclusion. Based on the results, you reassess your hypothesis, whether it is accepted or rejected. Selalunya, kalau yang dalam buku, sebab kita sudah tahu dia punya keputusan kan. So, we will rewrite the hypothesis, menulis semula hypothesis, dan kita tambah satu lagi ayat pendek, hypothesis is accepted. Okay, and that's all about it. Now let's look at the summary. Introduction to biology and lab rules. So you have fields and careers related in biology, safety and rules, communicating, and also scientific investigation.